Welcome back. So today we're going to explain a lot about our senses. We're going to go into tasting, seeing, smelling, hearing, everything. So I'm just going to kind of dive right into it, but I want to start with some vocabulary. So first, we kind of have to think about receptors and senses, because our brain speaks in action potentials, right? Like if you've seen my other videos, our brain speaks with, neurons receive neurotransmitters and things, and that's how our brain speaks. We have different receptors. So, for example, our eyes, we have photoreceptors, and our taste, we have chemoreceptors, and our pain receptors, we have nociceptors, right? We have so many different types of receptors throughout the body, and so I'm going to kind of just dive into it. So first, let's start with the eye. I want to start with the eye. It really doesn't matter where we start, but so the eye is everything about light and nothing about really anything else. So let's think of the eye. If I were to take the eyeball and cut it in half, so it's a hemisphere, and then put the hemisphere into the board, this is how it would kind of look like. Again, I'm not an artist, and this is probably not going to be 100% anatomically correct, but this is kind of what our eye looks like. Kind of. And then we have like this. So this is full fluid, and erase this actually. This connects. Sorry about that. Connects. It's full fluid, and then we have this thing. I'm going to draw it in blue here, a lens right here. And then we also have, I'm going to draw it like in red right here. This is our retina. So let me write retina. And like all these different things. So the lens, I'm just going to talk about two little different types of lens. Oh, also there's with the lens, there's these call them spindle fibers, like this. They go like this, and these spindle fibers are attached to muscles. And again, I could tell you all the anatomical variations and things about the size of the muscle and things like that, and what the muscle's called, but really I just want you to understand retina has the photoreceptor cells, right? And photoreceptor cells, there's rods and cones in the photoreceptor cells, and so so rods and cones. So rods, night vision, cones, day vision. So what we can see here, though, is that the muscle is attached to these suspensory ligaments, these ligaments that are attached to the lens. And there's two types of lenses. There's more than that, but I just want to generalize the lenses a little bit. So if you have a more bulb-shaped, drop-shaped lens, that's close vision or near vision. And then if you have a lens like more tight, it's far vision. So dependent on what this lens is shaped like is dependent on how we can see things. And so this part people get a little bit confused about. I've met several different people and students that get very confused about this thing. So obviously, because the suspensory ligaments are connected to it, the suspensory ligaments have a thing in it. So I don't know why I erased that, but here's this. If we have this is pretty trash, actually. Buckets. If we have these two, then the suspensory ligaments right here are obviously tight, and the suspensory ligaments right here are more loose. So the muscle has everything to do that, but it's not what you think. Like right here, you might be like, oh, maybe it contracts, makes it tight. Incorrect. So what actually happens, we have muscle. First, we have this muscle. And again, this is a cross-section of the muscle, right? The muscle, let's say it's a rod, cut it in half, boom, cross-section of the muscle. The suspensory ligament is tied around the muscle, kind of like this. And then here's the lens. So when the muscle gets relaxed, it's like, think about if you're in a chair and say you have some overhanging belly and you relax and it goes, right? It kind of grows bigger. That's exactly what's happening. So when it's relaxed, in the relaxed stage, it actually tightens the suspensory ligaments, which makes this tighter, which makes it so we could see farther away. But when you need to focus up close, this actually tightens and makes this looser. So that's why, like, sometimes if you zone out or something and you kind of just let all your muscles relax, those suspensory ligaments can become loose or become tight, and then you could see far away. So that's kind of a little bit about the eye. But another thing I want to explain about the eye is the retina and the photoreceptor cells, the rods and the cones. So if we think about light, light comes in. I'm going to draw light in two phases. It comes in and bounces off the lens and shoots right into the retina. The retina is just full of these cells, these rods and cones, and there's a ton of different research out there about the number of rods and cones, but I just want to talk about this is the small specifics of them and different things. So I'm going to have to erase all this. And then we're going to have to look into rods and cones close up. So I'm just going to call the rods and cones photoreceptors, right? Because it's photoreceptors, rods and cones underneath, right? Photoreceptors is a more generalized term. So right here, we have, I'm just going to draw the box because it's super easy. Photoreceptor cells. Then, so I'm going to just do PHR, photoreceptor cell. See? Then we have a bipolar cell, EP cell. And then we have a ganglia cell. Now, they have a significant amount of different things going on within these cells that help it so things happen. So there is this there's this thing called the G protein connected to the photoreceptor cell. I'm going to just draw it like this, and then it has the alpha, beta, gamma, right? But we just need to focus on the alpha, and then it has phosphodiesterase connected also to here. And so I'm just going to draw that right here and call it PDE, phosphodiesterase. So what happens when light hits, because that's how we see is light, we have a photon of light comes in, activates this G protein. G protein then eventually does gamma, beta, alpha. Alpha gets activated, goes into here, and then it activates phosphodiesterase. Now phosphodiesterase takes cyclic GMP and turns it into GMP. Just kind of takes off the cyclic GMP, and I'm going a little fast right now, and I'll go a little bit slower. But something that we actually need to see here is this thing called retinol. So there's 11-cis retinol and trans retinol. So I'm going to draw it a little bit simpler because the actual whole mechanism or chemical molecule of this chemical is pretty long, and I could draw it organic chemistry wise, but we're just going to have a hexene ring right here, and it's going to have a double bond right here. So a hexene with a double bond, there's some methyl groups coming right off it, but it also has a carbon chain coming out. And this carbon chain goes on for a little bit. Again, don't count the carbons on this or anything. That's not what's important. What's important is this bond right here. This double bond right here. These are all double bonds as well. And there's, I think, a methyl group coming off right here too. And there is a one of these groups coming off right here too. But this is what we need to think about first, is this is 11. Sorry. This is 11-cis retinol. So what happens with 11-cis retinol is we're looking into here, but kind of very close up, because this is a molecule, right? Molecules are super tiny, and sometimes you can put a methyl group right there. I'm just going to use red. Light comes in, hits this double bond, and then changes the configuration of the molecule. And so what happens is, if you know any sort of chemistry, this cis, and then the configuration changes, it will change into this. And so for those that might not know chemistry, organic chemistry, this might not have looked like a crazy change, but if you did pay attention to the placement of the hydrogens here, now we're not at 11 cis retinol, rather we are at 11 trans retinol. And there we go. So this configuration changes everything. 11 trans retinol helps with this alpha going in and changing this, or helps with the phosphodiesterase and everything. So I do want you to remember the cis to trans retinol. So now what happens is cyclic GMP comes in, and we have these little, I'm just going to call them like, we have these things, these bubbles almost, and they're filled with a neurotransmitter, right? When I explained neurotransmitters to you earlier, filled with a neurotransmitter called glutamate. Now glutamate comes in, but the problem is GMP inactivates, or GMP, what happens is it hyperpolarizes the cell. So the cell becomes, the photoreceptor cell, whether that's a rod or a cone, hyperpolarizes. And I forgot to draw it here, but there's a voltage gated potassium channel. And what that does to the voltage gated potassium channel is it closes it, making it so potassium cannot come in. Because of that happens, hyperpolarized glutamate cannot be activated. Glutamate does not synapse into the bipolar cell. But we have this thing here, I'm going to draw it in green, and it's supposed to, it can receive glutamate. And this thing is called mGluR6. mGluR6. But, so glutamate is not released within, it does not synapse to mGluR6. And what happens is we have right here a voltage-gated calcium channel. Because nothing really happens to the cell, it doesn't close this channel at all. So because of concentration gradients, calcium floods into the cell. And guess what? The bipolar cell also has these things right here that are full of the same neurotransmitter, glutamate. But because this happens, hyperpolarization does not happen. And now it gives it the ability to synapse. So we have glutamate synapsing into the ganglia cell. Now, because ganglia cell happens, we have this other thing right here. Sorry, I'm reusing colors, but we have it. It's called AMPA, A-M-P-A, AMPA. AMPA gets activated by the glutamate, but what actually happens with AMPA and the glutamate activating is we have these other channels. We have, again, a voltage-gated calcium channel, but it activates this voltage-gated sodium channel. These flood into the cell, and then glutamate is released again. And there is, I don't know if you could see this, a neuron right here. And an action potential, as glutamate is released, an action potential is released to the brain. And it's saying light is happening. So, like I remember at the very beginning of this video, I don't know how long ago that was, but we had the different types of receptor cells. We had photoreceptors, we had chemoreceptors, we had nociceptors, all those different types of cells that go on. So dependent on the cells, it goes to a different part of the brain. So this goes to the part of the brain that receives light. And this is what happens here. So, but I kind of need to explain a little bit more in depth of some other things. Okay, sorry, I just got some water, but so what actually happens here, so let's just kind of go back a little bit. This is a photoreceptor cell, whether or not this is a rod or a cone. So a rod or a cone, we could let's just go to on, center, off, surround. So our cells are kind of built up like, I don't know how to explain it, but let's just say this is the photoreceptor cell, right? And the photoreceptor cell has a ton of just different cells in like this. And they're more minuscule than this, but not. This is fine. So we have the outer ones and we have the inner one. Ones that are more outer and ones that are inner. And so we'll call it center. So this is the center and out of the center. So dependent on the cell, some cells get activated by darkness and some cells get activated by light. Remember I said rods and cones, the difference between rods and cones, night vision, light vision, day vision, whatever you want to call it. So sometimes we have a cell here, and say the cell is actually isn't able to hyperpolarize because the photon of light does not come in. So what actually happens is glutamate is released here, releasing mGluR6, which does not release glutamate here, and AMPA is released. So what happens here is it's opposite, right? And so this helps us see edges. Something our minds do so well is seeing edges. Our eyes do so well seeing edges, and that's why we can see in such high definition. It's because of the edges of things. Whether or not if you're whatever you're watching this on, whether it be a phone, a computer, or your TV, it probably has some edges around the corners, and we can see those edges so well. If you look up and you see a bookshelf, you can see the edges so well, and that is because of this on-center, off-surround. And so I'm going to explain it kind of just like this: on-center, off-surround. So, for example, I'll just do light with green. This gets activated by light and this gets activated by darkness. If light hits like this is activated and this sends action potentials, and this sends action potentials because it's darkness, but this does not send action potentials. And you're like, this is the most confusing thing ever. And it kind of is, but I'm going to kind of explain it with some simple math. So I'm going to label this as 2, this as 2, and this as 6. Because I want you to know that the center cell sends more action potentials than outer cells. So total possibility of this whole thing, if somehow this was receiving light and these were receiving darkness, could be 10 action potentials. But right here, we're sending 8 action potentials because 6 plus 2 equals 8. 8 action potential because this is receiving darkness. Now, if you ask, well, what if the whole thing was covered in light? Then we're only sending how much? 6 action potentials because on center, off surround. Now, say this was darkness, the green was darkness. I'll just erase it to make it easier. So yeah, say we got hit with darkness. There's no more light. How many action potentials are getting sent to the brain? 4 action potentials are getting sent to the brain because on center, off surround, this gets activated by darkness. So it actually turns on and this turns off. So we're not sending 6, we're sending 4. So that's why it's kind of harder to see at night because less action potentials are being sent to the brain. So this is the eyes. So if you have any questions, comment below. That's what's going to go on. So now I'm going to explain, what am I going to explain? I'm going to explain smell and taste, but I'm going to get some water first. Thank you for letting me get some water. So now we're going to explain, let's explain, let's do smell. So smell, we have a different myriad of things, but we kind of first need to think about the nose and the mouth and a little bit of the neck area, if you will. So this is going to help us a lot. I'm going to draw a couple of anatomical things that's going to help us to understand what's going on, and it will help us understand. Okay, so again, I'm no artist, but this is the best rendition of what I'm going to do. Right here we have our guy. And our guy we have a couple things go on, right? We have our nose here. But our nose kind of goes like this. Again, this is very simple. Very, very, very simple. And this is just kind of how I'm just going to fade it up in there. I don't know. But this is just kind of what we're going to do right now. We don't really need to know a ton of anatomy, but yeah, this is what's going to happen. We draw some teeth right here, right? So we have our nose and our mouth and everything. So what we really need to think about anatomically, this is actually important anatomically, is the olfactory epithelium. The olfactory epithelium sits right up here, right on the top of your nose, your sinuses, and your brain is literally like right here. So we have, obviously, we have like a bone right here, and that bone's called the cruciform plate. But I'm gonna kind of draw what's going on here. So again, we have that olfactory epithelium, and so I'm going to redraw the olfactory epithelium right here. Olfactory epithelium is like this, and then we have this the plate right here, the bone plate. I'm just going to call, you literally don't even need to know that this is called the cripiform plate, but you could just know it's bone. And it's a special type of bone, it's a little softer. And then literally right here is the olfactory bulb of the brain, right? And again, what did I say earlier? The brain speaks in action potentials. And so we have to change our senses from whatever they are to action potentials. So for smell, we have to change them from the particles and things that we're smelling, the aromatic rings, if you will, that we're smelling, and turn them into action potentials. So how do we do that? So we kind of need neurons, right? So I'm going to just kind of draw neurons like this. And we'll zoom it in right here again. So they branch out in here in this area, and then they kind of come out like this, and then they bundle up or they synapse past the plate, and then they go into the olfactory bulb. And in the olfactory bulb, that's where like they synapse with other things and they release neurotransmitter to help us sense what that smell is. So I know I keep zooming in, but we're gonna have to zoom in again. Let me zoom in right here. So if we have our thing like this, so those things that come down, they grow back and they're very important. And we kill them off a lot, but they're very important for smelling things. They're pretty much just like hair type things. But sometimes if you get in a car accident or something, you get your rear end someone, your head could hit here, right? And it could break this and it will actually chop off all these nerves. And if those nerves are chopped, then you can't smell ever again. So like COVID-19, the COVID-19 virus killed these. And so that's why, well, it depended on the variation of the virus and things. So that's why a lot of people couldn't smell or taste for a long time with COVID-19 because they destroyed these. But again, like those people can smell back, hopefully. If you can't smell, I'd probably go see a doctor or something. But let's go back to here. So this is this, just zoomed in. Like I explained before with the eyes, we have this thing called the G protein. And it's about the same, but I drew this one a little bit smaller. So we have an activator coming. And like with the eyes, we have a photon. This is a different type of chemical. And it activates adenylyl cyclase. So we have cyclic AMP, go to AMP. What that does, so first we have this happen, then this happens, and then it causes there's these voltage gated potassium or calcium channels, and it causes calcium to come into the cell. Calcium comes into the cell, and as the positive charges keep going up and up and up, there's already sodium within the cell. Sodium's like, okay, this is annoying. Sodium leaves the cell. As sodium leaves the cell, I guess it's really just chlorine that leaves the cell and sodium comes in, sorry. So positives come in, negatives leave. This creates that kind of length constant, that belong that I talked about in my earlier videos, creates an action potential that goes right to the brain. Sorry, someone just called me, but back to what I'm talking about. So we just talked about that's how you smell, right? Very important. This is how these chemicals that we smell in, these aromatic rings that we smell in, right? Aromatic range. But these things that we smell in, how they change into action potential so our brain can interpret what we're smelling. And smell is like one of the most powerful. Oh, I didn't explain that either yet. But anyway, smell is like one of the best things that we have for memory. We could always smell things. So, but fun fact, this is the epiglottis. Because this goes to the stomach and this goes to the lungs. The epiglottis is a muscle that kind of holds this, and that's how we laugh, actually. Not crazy important information, but when we laugh, it's the difference between the air getting pushed up from our stomach and going through our lungs, and it's moving, and that's how we laugh. But let's go into taste buds. Okay, so taste, again, it's the same thing. We need to convert whatever molecule into an active potential so our brain can know what's going on. So if we have our tongue, I'm going to draw a massive zoomed in photo of the tongue. It's split in half, so we can see it on the board. We kind of have these things like this. It looks horrible, right? That looks horrible, but those are called papillae. And these papillae at the end are these kind of like divots. Like you could literally look at your tongue and see these little bumps and grooves. But at the end of these, we have these bulbs. They kind of look like light bulbs almost. I'm going to draw them like this. And these are called taste buds. So this is what's going on. You've heard about taste buds before, I'm assuming. And the taste bud is the taste receptor. That's like the cell that's recepting everything. So, well, kind of. The taste bud is full, I should say, of taste receptor cells. Right. Full of taste receptor cells. So again, I'm going to probably erase all this. Just remember this map. I just kind of want to draw this map so you could understand. When you look at your tongue, you see all those things. That's called papillae. And on the papillae, you can't see those, but they're taste buds. And underneath the taste buds are taste receptor cells. And again, I'm just going to draw the taste receptor cell like a square. So much easier for me to draw it as a square. There's a couple things that I need to explain with taste, and because there's different types of these cells that get different types of things. So we have salt, sour, sweet, bitter, and umami, right? We have those types of things that are in our tongues, and you can look up online like the maps on your tongues and things, and depend on whether or not you're eating different cultures of cuisine will help you feel those different types of things, whatever it's spicy or what it's sour, whatever. So like for example, salty, you're feeling ions like sodium. Sour, you're doing hydrogen's acidity. That's why things are sour and stuff. Sweet, so that's kind of what's just going on here. So in all these cells, we have this anatomical variation between them, slightly, but in all our circoplasm, we have it's just full of calcium. Sorry, we got full of calcium, and then underneath here we have a neuron, right? And the neuron is like the subway that carries the action potentials to the brain. this neuron or this cell has a ton of these, again, these bulbs that are just full of, I have to erase this section, full of the neurotransmitter glutamate. This whole video, glutamate is all over the place. All over the place is glutamate. And glutamate is so important, this neurotransmitter. So anyway, this is kind of the anatomical variation between the two. So, and then they have a voltage gated calcium channel, and they have a voltage gated potassium channel right there. And it just depends. And this, has receptors and other cells and things. But it depends on whether or not it's a salt, sour, sweet, bitter, umami. So the salt and the sour have two different types of cells. And then sweet, bitter, and umami actually have the same cells that just receive it differently. So what I'm trying to say is that they work the same. But depending on what comes in is what you taste. Umami, bitter, sweet, or sour. These work the same, these work different. So let's go into salt. So salt, for example, has, because it's sodium, has a sodium channel. Sodium comes in, and it's called an epithelial sodium channel. I'll write that right there. ENAC. So epithelial sodium channel. And what happens? Sodium comes in. Sodium's positively charged, and so sodium keeps coming in, positive charge, and it sets the length constant out. What the length constant does, it activates these things, and guess what happens? Glutamate's released, and active potential sent to the brain. Okay, so if you're taking notes, you're going to probably have to write this 5 different times, or 3 different times, but I have a whiteboard. So sour, again, it's the acidity. So acidity is based on hydrogen ions. Hydrogen comes in, and what actually happens is it blocks this voltage-gated potassium channel, and that allows not potassium to come in, and kind of nothing really happens to it. Because the smooth endoplasmic reticulum, I don't know what I called this earlier. Hopefully it was SER, smooth endoplasmic reticulum. But nothing really happens, and that's how our hydrogen comes in, same thing happens, and actually potential sent, we're like, oh, that's kind of gross, that's sour. So now let's explain sweet, bitter, and umami. So with sweet, bitter, and umami, it completely works different. So we have a G protein. And I know I've explained G protein a lot in this video, but again, everything really has a G protein, and it's very important. So G protein comes in. So whether or not it's carbs, calcium, or ketone, I'm pretty sure it's both ketone and all, comes in, activates the G protein. And then what actually happens there is phospholipase C is activated, and IP3 comes out of phospholipase C. And what happens with IP3, we all know with this smooth endoplasmic reticulum, it kind of has this thing go on, kind of activates the channel. The channel gets activated, and then calcium floods into the inside of the cell. And then what happens there is the voltage gated sodium channel shoots sodium out. This creates a different action potential. Neurotransmitters released and goes down the neuron. And now we have taste. Now you know if you're tasting sweet, sour, bitter, umami, whatever it may be. I've been explaining a lot right now, and I'm going to get some water, and I'll be right back. Okay, so now I want to explain sound. Sound is actually quite intense. I feel like I explained the eyes, and the eyes are pretty cool, and there's a lot going on with the eyes. and probably a lot of confusing things that I said. And then I explained smell and taste. And I was like, oh, okay. But now we're going to explain sound, and sound is significantly more intense. So we're going to go into a lot of anatomy, a lot of obviously physiology, maybe a little bit of chemistry, many different things. So first, I want to do a little bit of anatomy. And this anatomy, let's just do the ear, right? We hear from the ear. Definitely cartoonish here. All right, but bear with me. We have the ear, and then right here, with the ear, we have this thing called most of you already know, eardrum. We have an eardrum, and then we have three bones. And these bones are actually important. I'm not going to say the names, they're crazy names, but let's just say three bones. And the bones are kind of like triangular, kind of, and then there's just one bone like this. So 1, 2, 3 bones. And so again, the whole thing with hearing, with smell, with taste, with whatever it may be, we need to send whatever external factor, whatever physical thing into an action potential so the brain could sense what's going on. So that's where this comes in. So we have sound waves. I think hopefully a lot of people know sound waves. Maybe if you ever see a jet, like sound waves are really cool and they're legitimate things. Can't really see them that well. Maybe you can again with the jet. But sound waves come in, and that's what the whole ear anatomically is built for. So we could retrieve these sound waves. The sound waves hits the eardrum, and the eardrum starts to oscillate like this, starts moving in and out. And then I'll kind of shift these bones around. It's kind of insane to think about, but I'll shift these bones around and move these bones. But there's another anatomical thing that I have not drawn yet, and that's called the cochlea. Maybe you've heard of the cochlea before, maybe you haven't. That's totally fine. The cochlea is kind of this organ in the back, or I don't know what you want to say, but it's definitely is wound up like this a lot. But I'm going to draw it less wound up so that we could kind of understand what's going on. So we're going to have it go like this. Cochlea, right? I could draw it. Again, don't criticize my spelling. I'm just here for physiology. So we have the cochlea. And the cochlea has kind of this little thing going on in here like this. This is drawn very dramatic. Usually this is a lot closer. So we have this thing right here. Again, it's usually drawn a lot closer, so it doesn't have to be that big, but this is called a round window. And I'm sorry, this little part of the lecture has been all anatomy, but there's so many different things that's going on. And I have to keep spelling things. Okay. And then we have this thing. I'm sorry, I'm going to use red twice, but it's pretty important that I'm going to use red twice. Or this part, like we all know the eardrum. This part is more important for this lecture. Basilar membrane. So this is what's going on. So the whole cochlea is full of, I guess I could use blue right here, perilymph fluid. It's all full of perilymph fluid. And so this is how it's turning into an action potential, right? So we have the eardrum, sound waves go into the eardrum, oscillates the eardrum, moves these bones around, and then it kind of starts moving around the water. And again, the cochlea is wound up a ton, and so this basilar membrane is wound up a ton with it. And so the basilar membrane, we could kind of, if we took the basilar membrane out and unwound it, kind of like a sleeping bag, and look down on it, it would kind of look like this. And I drew that like this on purpose because it gets bigger as it goes more into the convoluted everything. I don't know what you want to call it, but the coil, if you will. And so the bigger you get, and so if you kind of imagine when you open up a piano, there's a ton of different strings in that piano. So many, right? And so think about that same with the basilar membrane. But the closer you get here, the bigger, or the increase in number of hertz, and over here, the smaller number of hertz. This is how our brain can hear things. So like again, if you get older and older, these become to be getting more stiff because the vibrations don't work. And so eventually this stops working as much, the higher intensity of things. But we're now going to go zoom in a little bit to the helicotrina. And the helicotrina, and like zoom in again, we're going to go in and cut it in half, just like I did before. So remember this thing, if you're taking notes, make sure you have this drawn. Pause the video, you could draw it. But I'm going to erase all this. So we take the whole thing and we're going to make a big, big circle. Right? This is cutting the helicotrina right in half. And this is the artist part, right? And I'm not the best artist, but I'm going to try my best to really just get it going here. I need to see if you guys can see. Okay, so sorry, my screen was closed. I just had to open up the video again. Helicotrina, and then we also have the striae vascularis right here. Striae vascularis. And there's a ton of different anatomical things going on here. So we're going to have just kind of this line here. We'll draw it kind of thick. And I'm just going to call things just letters because it's going to be significantly more easy. If you want to actually know the anatomical name for these things, you can. But if it's just SV within the cochlea, that's fine with me. So that's what we're going to draw. SV and then the cochlea. That's like the section right there. And then we also kind of have this thing going on here. It's like, and that's called the ST. I have this thing right here. It's called the tectorial membrane. So this is kind of what's going on within the ear. We also have, because we have different sections of things, and it's just different fluid concentrations and different areas of things. But right here is called endolymph. And I'm sorry if you drew in pen, but it's a lot easier for me to draw it the way I did, like this. And so we have the endolymph and the perilymph and things like that. But this kind of moves out like this. So endolymph goes out, and this is kind of like a C shape right here, all the way around. And then those are the striae vascularis, right? Within the endolymph, we have these little super minuscule hairs. And those super minuscule hairs have hairs on the hairs that are even more minuscule, enough so that it could sense atoms. That's how minuscule these hairs are. And so I'm going to kind of draw it a little SpongeBob-like. I'm going to draw it a little bit like, let's see here, okay. Okay, there's a reason why they're decreasing and things, right? So I'm going to draw this, but over here, and these are the hairs that could touch atoms, okay? Into the tectorial membrane, right? And then there's also another anatomical thing that I completely forgot to draw, and that's 100% my bad. But it's like this. And then we also have, I'm keeping with the same color, red. Remember, red meant basilar membrane. So we'll just have the basilar membrane right along there. And so actually these keep going. There we go. So those are the hairs, and they're all over the place, but I just draw two because I don't want to draw them all, right? So there's so many things that go on within the cell. Let's start here in this striae. So on the complete outside of the striae, we have, well, first we have the sodium potassium pump, right? And the sodium potassium pump is, like I say always, like one of the most important pumps of all time. The sodium potassium pump creates the concentration gradient, creates the electrochemical gradient, and we have this other special type of pump. And this is more like a leak channel, and because of this gradient that's created, we have several things come in. We have potassium come in, we have sodium come in, and we have calcium come in. All those things come into the cell. We also have, we have these things come in. Sorry, I don't know why I wrote calcium, but it's sodium that comes in. And just like any other cell, there's leak channels everywhere, right? But the striae vascularis is actually, like, I think one of the most full membranes of leak channels in our whole body. And so we have so many different, like, potassium leak channels and things, and chloride leak channels. So when this concentration gradient actually starts, we have potassium leave the cell. I drew this on the wrong side, I'm sorry. Potassium leave the cell and chloride leave the cell. And remember, what happens when chloride or potassium leaves the cell is it leaves behind negative charges. Now, these negative charges are pretty important because they help push things along. And so with the striae vascularis, the same thing happens here. Another concentration gradient's made, and potassium is actually able to end up in the endolymph. So eventually potassium ends up in the endolymph, and it creates this positive charge. So what we really need to see in the ST, we have a very high concentration of sodium. And obviously now we have potassium coming in. The thing about the high concentration of sodium is it pushes sodium out. What did we learn before? Is that when sodium leaves, it leaves behind a negative charge. This makes this basilar membrane part highly negative, and because of all the potassium, it makes this highly positive. Now there's a gradient. This gradient helps it so that potassium can come in and wants to go into here because it wants to go to the negatively charged things. And there's also a low concentration of potassium there. And so that's kind of what's going on right now with the Katrina and all these leak channels and things, the endolymph. And so I want to kind of zoom in because you're like, well, how does this do really anything to the action potential sent to the brain? And that's a very good question. And so that's what I'm going to explain here with this hair. So I'm going to erase this arrow. And now the hair is actually kind of cool to look at. I think I'm missing a color. So with the hair, we have these potassium gated channels. Potassium gated channels, and these potassium gated channels, they have this thing called they're just like these proteins, and so these proteins, it's like protein tethered to the hair. And so if we kind of imagine these hairs, and we kind of imagine, say, a windstorm, if you will, the hairs kind of move back and forth. And when they move back and forth, this protein tether, that's connected to this potassium channel, it will open the gate. It's a gated channel because once it opens the gate, obviously, there's potassium on the outside. Potassium comes into the cell, so gate's open, potassium comes into the cell. Like I said before, right? Potassium comes in. Potassium comes into the cell, and then we have a potassium leak channel here, and it leaves the cell, and we'll kind of just think right here is the bacillar membrane and things. So now potassium can just travel from all the way outside of the cell to here. It's pretty cool. So all like the blood vessels and things are out here because this is so specific that if we had blood vessels here, we'd be able to hear red blood cells bouncing off each other. And so all we'd ever hear was over and over and over again. So that's why all the blood vessels, all these things happen here, all these ion movements happen here, so we don't hear it. And then when you come into here, we can hear it. And again, so this, because the potassium leaves, we have this kind of drops off over here, right? Again, this is not anatomically correct or anything, but we have another potassium sodium channel. And so as potassium leaves, because it wants to leave because of the negative thing, it creates another concentration gradient. And this happens, and then we have these, and they're also full of neurotransmitter. Because of the concentration gradient and things, and we have a voltage gated calcium channel. The neurotransmitter is able to get released, and just like everything else, what we have here, we have a neuron. And an action potential is sent to the brain. Again, dependent on the bacillar membrane, we have different hertz. So it depends on depends where you're at in the bacillar membrane. Remember that drawing I drew that depends different hertz. That's why you could hear tones. That's why older people can't hear certain frequencies because those have become stiff and broken and things. So this is how we can hear. So with hearing, let me go back a little bit. We have the ear. Sound waves come into the ear. As I'm speaking to you, sound waves are going into your ears. The sound waves go and hit the eardrum. The eardrum oscillates and moves those bones, and it could push. It's like if you took a water balloon and you just poke the water balloon, that energy is going to go everywhere. That happens within the perilymph fluid. And if we zoom in onto the helicotrina within the cochlea, this is the photo we get. And then we have these striae vascularies that come in, and they have all these different channels. Ion gradients could happen. Hairs get vibrated because of the different vibrations of what's on the bacillar membrane. Different concentration gradients and things happen. All eventually leads, because of movement and concentration gradients, we could have neurotransmitter release go into the neuron and tell our brain, hey, you're listening to music. So that is the ear, that is taste, that is smell. I know this video is probably an hour long, looking at the time thing. Yeah, sorry about that, but it's pretty cool stuff. So I'll see you next time.